Okay, so we talked about the urinary system, the kidneys, the ureter, the bladder, and the urethra. So how is urine formed? So we're going to get into the kind of the mechanism of urine formation, and then we're going to talk about urine modification, and then the roles of the kidney in regulating other things as well. So, see if I lost my pen. Seems like I lose it every time I change. There we go. All right. So if you'll remember, the kidneys are going to filter your blood. And when I say blood, I really mean the plasma. So it's going to filter the entire body's plasma 60 times a day. So your blood is going to go through your kidneys 60 times a day, and that plasma, the watery part, is going to be filtered 60 times a day to get rid of metabolic waste, excess ions, and water. Or maybe it's going to hold on to ions and water. It kind of depends on what your body needs. Your kidneys can respond to either getting rid of excess water or holding on to water if you happen to be in a dehydrated state. So when that blood passes into the kidney, what you're going to form first is called filtrate. So that, if you remember from the first video, when the blood goes into that glomerulus, right? So the blood goes into the glomerulus and then something goes out into the proximal convoluted tubule. That's called filtrate. And it's really the kind of the watery portion of the blood. So it's blood plasma minus proteins. So the big proteins don't pass into the filtrate. Ions do, water does, sugar can, glucose, but your big proteins won't cross into the filtrate, nor will red blood cells or white blood cells. So your cells don't leave either, all right? So just kind of the watery part goes into the filtrate. So you don't want to get rid of all of that. If your plasma is going to be filtered 60 times a day, and all of that went into your ureters, into your bladder, and out through your urethra, you would dry up like a prune, okay? So we know the kidneys must do something to get most of that back. Even though we're forcing all of the kind of liquidy part of the blood into the kidneys, we're going to have to resorb a lot of that, otherwise we would dry up and blow away. And so the actual urine has been processed and most of the water taken back, so you really only get rid of about 1% of, or less than 1% of what is filtered every day. So less than 1% of that total filtrate is actually going to be taken out of the body as urine. And in that urine, you're going to have your metabolic waste and anything that's not needed. Okay, and that can be excess water, it can be excess ions, it can be other metabolic waste like urea and things like that as well. All right. Well, it takes 3 steps to make urine. There's something called filtration, reabsorption, and secretion. Okay, so filtration, reabsorption, and secretion are the 3 steps. Now, filtration occurs in the glomerulus, so we call it glomerular filtration. So we're trying to make it easy on you, telling you where it happens. Reabsorption and secretion happen in the tubules. Remember the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule? This is where your reabsorption and secretion is going to occur. So what reabsorption means is that filtrate, you're going to get the good stuff back. You're going to get the glucose back, the amino acids back, most of your water and your salts, and any other thing that you need to survive in your blood. The good stuff is going to come back and be reabsorbed. Secretion is when you want to get rid of stuff. So if you're going to take stuff and add it to the urine. This is stuff that was not in the filtrate. You're adding additional stuff to the urine. You're secreting stuff into the filtrate to get rid of it. Okay? So those are the 3 steps. And they take place in different parts of the nephron, in the glomerulus, and then in specific portions of the proximal tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct. And we're going to show you where all these different steps occur in detail. So what is glomerular filtration? It is a passive mechanism, which means it does not take ATP. Okay, it's passive, where you're basically filtering something using pressure, hydrostatic pressure, which is water pressure. So you're basically pushing stuff through to be filtered by just using water pressure. So imagine if you had maybe a sieve, and think about coffee almost, like when you pour coffee and you put the coffee grounds in there, then the water is going to fall on top of the coffee grounds, and as it's pushing down, it's pushing the coffee out the other end. Kind of the same idea. So it's being pushed by hydrostatic pressure. So this is going to happen in the glomerulus where filtration occurs because it's a very efficient filter. It's very permeable. It has a large surface area. So if you remember that arterial and you have that glomerular capillary, so because it's very dense capillary bed, there's a lot of surface area. There's many, many, many glomeruli inside of a single kidney, and they're very permeable, and that allows for a lot of filtration to occur. It's also true that the blood pressure in the glomerulus is a little bit higher than you expect in other capillaries. So if you think back to blood flow, remember we said you take blood from the aorta to the arteries, and then to the capillaries, and then to the veins, and then back to the vena cava. Remember, the blood pressure was high in the aorta, right? And then it kind of drops to almost nothing in the capillary beds, and then it kind of stays that way in the vena cava until it gets back to the heart. So your blood pressure is arterial pressure. Well, this first glomerular capillary is still on the arterial side, all right? So it hasn't gotten to that zero capillary pressure yet. So there's actually pressure in the glomerular capillaries that helps push that filtration as well. When this filtration is occurring, big stuff won't pass. So there's actually a molecular sieve that won't allow things like proteins. So things that are bigger than 5 nanometers are not going to filter, like plasma proteins and things like white and red blood cells. Okay, so just the small stuff gets through. And if you remember back to blood pressure, and we talked about how things move in and out of a capillary bed, we talked about osmotic pressure and how hydrostatic pressure kind of pushes things out and osmotic pressure sucks them back in. By keeping these big proteins inside the blood, that's going to help suck stuff back in into the blood when we talk about reabsorption. So these big plasma proteins are really going to be what give us our osmotic pressure, the sucking pressure. So I know you guys hate it when you have to remember stuff from previous units, but this is one of those units you're going to have to go back and think about how blood flows through the arteries, to the capillaries, how exchange occurs in those capillaries, because the same thing's happening here. So this is not something you can just go, oh, that was way back in Unit 1, I can forget it. Nope, you got to bring it forward so that you can understand this unit as well. Sorry. We're building. It's a building semester, much different than 201. All right, so let's look at this glomerulus, okay? So here's our blood's coming in. Now, let's see if I can change my color so it's a little bit easier to see. It would be maybe this color. Yeah. So remember we said the nephron has 2 capillary beds kind of in a row, and the first capillary bed is called the glomerular capillary, okay, so the glomerulus. So that's all this stuff in here, okay? And so blood flows in through the arteriole and then forms this capillary bed, and then it flows back out, okay? So blood flows into the capillary bed and it flows out, just like you're used to. Blood's got to flow in one direction. Well, blood enters into the via the afferent arteriole, and it exits through the efferent arteriole. So afferent takes the blood to the glomerulus, efferent takes it away. And while it's in the glomerulus, this is where you form the filtrate. This is where the blood is filtered. All right? So you have your afferent arteriole, which is going to be in a bigger diameter than the efferent arteriole. Okay? You have your glomerular capsule. So this glomerulus is just kind of inside of a bag, if you want to think of it as the glomerular capsule as a bag, so that you can force the filtrate into this space, okay? Then the filtrate will then exit through the proximal tubule and then go through the nephron, okay? Now, when the proximal tubule does its little thing, then it makes that long loop of Henle, and it comes back up as the distal convoluted tubule. That's a D. The distal convoluted tubule comes back to its own glomerulus, its own efferent arterial glomerular area, and we call that the juxtaglomerular apparatus. It's where the ascending limb and distal convoluted tubule, kind of that transition point between ascending limb and distal convoluted tubule. This is that portion of the tubule, and it comes back to its own afferent and efferent arteriole, and it actually has little cells associated with that are sensory cells, and we're going to talk about that later, but these play a role in monitoring salt and water balance, and we'll talk about how that works later. So you have your juxtaglomerular apparatus, which a lot of times I'll say is called your JGA. You've got some sensor cells called macula densa cells, and then you've got some things called granular cells and mesangial cells. We'll talk about those a little bit later. What I want to focus on right now is the filtration part, because we're talking about glomerular filtration. And you'll notice as the blood goes to the afferent arteriole, it's going to then go through all these little capillary vessels, all right? And these capillary vessels are covered in cells called podocytes. And so these podocytes act like a sieve, and they allow the plasma to go out, but makes the protein stay in. So they're that molecular sieve, that nano small filter that won't allow the big stuff to pass through. So let me take you to a bigger picture. So you can see here's the afferent arteriole, the efferent arteriole. The blood's going through, and as the blood is being filtered out, these little podocytes kind of help hold things back in and allow for the water and the smaller ions and things to pass through, and the big things can't. So again, form follows function. So these are called podocytes. Here's the cell, and it has a little foot, so pod like ped, like feet, has these little long foot-like appendages that make little toes, if you will, that act as a filter. All right, so this is where it gets to be a little confusing for some people when we talk about filtration pressures. All right, so remember back to capillaries, where as the arterial pressure is dropping to just about zero, there's a little bit left, and that pressure pushes the stuff in the blood out into your tissue. So that's how you get the sugar, and that's how you get oxygen, and things start kind of get pushed out into those tissues. But then on the venous side, remember you have those big albumin proteins that stay in the blood, and they suck the water and the bad stuff, the waste products out of the tissues back into the blood. That's how it normally happens in a capillary bed. Well, it's pretty similar to that in the glomerulus. So you have more pressure than normal in a capillary bed. You have 55 millimeters of mercury, that's a pressure reading, pushing, hydrostatic pressure, pushing out into that capsule. So that's going to push all the liquid stuff out. But you don't want everything, if you had that much pressure and you were pushing everything out into the glomerular capsule, this side would be empty. And that's not the case. We know that there's going to be blood that exits the efferent arteriole. So there's got to be some pressure that's pushing back that doesn't allow all the blood plasma to exit into the glomerulus. And so the pressure that's pushing back is the osmotic pressure and the hydrostatic pressure of the capsule itself. Okay, so think about if you had a hose, okay, and so water was coming out of the hose, and you took another hose. So say this hose is blowing water out at maybe at 2 pounds per second. But this hose is blowing water back at 5 pounds per second. What's going to happen? It's going to push the water back into that hose, right? So you're going to have pressure. There's water inside the glomerulus. It's pushing back, and it's going to push some of that water back into those arterioles. But it's only 15, and so 55 is greater than 15, so that's still the net pressure is pushing out, right? But that's still 15, and 55 minus 15, that's a big pressure. You would still lose all of your filtrate, all your plasma is filtrate, and that's too much. You've got to have something pulling more water back in or sucking it back in, and that's the osmotic pressure. And that's the big plasma proteins that are inside the capillaries that suck, using osmotic pressure, suck the water back in. So if you have 55 minus 30 plus 15, means you have a net filtration pressure of only 10. So it's just enough outward pressure to push the filtrate into the glomerulus, but not so much that you don't have blood leaving the efferent arteriole. It's a little conceptually hard, so take a little time thinking about that, and so you can understand that everything that goes in doesn't all go through the filtrate. Okay, some of it is brought back in, sucked back in, so that it can then travel through the efferent arteriole, so that you have blood that can go to that next capillary bed. Remember, there's 2 capillary beds in sequence in the kidney. You have your glomerular capillary bed, and then you have those peritubular capillaries or vasorecta, depending on if you're in the cortical nephron or juxtamedullary nephron. All right, So one of the things you can do is measure how well kidneys do this, and we call that the glomerular filtration rate, or GFR. So I will probably say GFR from here on out because it's easier to say than glomerular filtration rate. And that's the volume of filtrate that's formed per minute by the kidneys. And a normal GFR is about 120 millimeters per minute. So if you do a GFR study and you want to see if someone has kidney damage, you would expect to see that GFR number go down. So that's kind of an indicator that maybe things aren't going very well with their kidney function. And it's governed or controlled by several things. The total surface area available for filtration. So if you had kidney damage and maybe your glomerulus was damaged, you would have less surface area, right? The membrane permeability, maybe you have scar tissue that's formed on the membrane, so that could change the filtration permeability. And the net filtration pressure. So how many proteins are left in the blood? What's your blood pressure? Remember, net filtration pressure is the hydrostatic blood pressure pushing out and the hydrostatic glomerular pressure plus the osmotic pressure pushing back. Okay, so if any one of these changes, that changes the net filtration pressure. The most obvious thing is if you have high blood pressure, that's going to increase the hydrostatic blood pressure and push things out. Okay. Now, we're going to talk more about that as well. We're just kind of getting all kind of laying the stage for how all this works. So not only do you have filtration, you also have reabsorption and secretion. So reabsorption, tubular reabsorption, occurs in the tubules, in the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule and collecting duct. You need to know what's happening where. Most of the reabsorption occurs in the PCT. And what happens there is you get most of your water back, most of your sodium back, all your nutrients, your sugars and things like that, small proteins, not the big ones, but the small proteins, and your ions, sodium, potassium, all your different chloride, calcium, just think of all the ions. You want to get all that back so you don't get rid of it. In the loop of Henle, specifically, you're going to start to concentrate the urine. So this is what we think about. If you look at your urine, sometimes it's kind of bright yellow and sometimes it's kind of a pale yellow. That concentration of urine is going to occur based on your level of hydration. And so in this area, you kind of focus on salt and water balance. So you're going to focus on reabsorbing or not reabsorbing water, and the same thing with sodium, potassium, and chloride. So the balance of those things will determine whether or not your water is going to be, your urine is going to be concentrated or dilute. The distal convoluted tubule and collecting tubule is where you have some more reabsorption occurring, and it's under the control of hormones, and specifically parathyroid hormone, which is going to help with calcium reabsorption, ADH, which is anti-diuretic hormone for water reabsorption, and then aldosterone and atrial natriuretic peptide for sodium reabsorption. So this is where your endocrine system kind of comes in. Again, that has to do with regulating water volume, which has to do with regulating blood pressure, and then cardiac output. So all this stuff is starting to tie together now, I hope. So when you're talking about how these things move back into the blood, so remember, this is that second set of capillaries that are going to be around the tubules, the peritubular capillaries or the vasorecta around the loop of Henle. Things are moving from the filtrate back to the blood, and they can move in several different ways. There's called the transcellular route, and that means it goes across the membrane, right? So it goes through the cell, so you have like water and solutes can go through the cells themselves, and then there's always going to be a space, an intracellular space, kind of interstitial fluid, if you will, between those cells and the blood, and it goes right into the blood. Then you can also have paracellular, which means it goes between cells. So where these cells are connected, there's going to be some spaces, some gaps, and so things can actually go between these cells into the intracellular or interstitial fluid and into the blood. I'm not going to read this. It's in your book, so that way you guys can read. So I hate to just read a slide to you. But it can cross either through the cells or between the cells from the filtrate. This is the lumen of the tubule. These are the cells that form the tubule. It's the interstitial space. Remember, all your body organs are kind of floating in this interstitial fluid. And then this is the actual peritubular capillary. All right, so then tubular secretion. This is where you're going to get rid of stuff. So reabsorption in reverse, if you will. So when you're going to get rid of stuff, what happens here is you're getting rid of things like potassium, hydrogen. So hydrogen, what does that tell me? What is that? What do I think about when I think of hydrogen? pH. So here's where you're going to start having some acid-base balance, and we'll talk about acid-base balance as we move along. Ammonia and urea are some metabolic byproducts that we got to get rid of because they build up of those in our bodies is not good. Organic acids, creatinine, so several different things we want to get rid of. We can also get rid of substances that have been bound to plasma proteins, so other things that are being carried through our blood we need to get rid of can be secreted into our urine. Excess potassium can be secreted here. And also we can regulate pH not only using hydrogen but also bicarbonate. So HCO3 minus is bicarbonate. And again, we'll talk more about that when we talk about acid-base balance in a couple of weeks or in a few more lectures. All right, so I like this particular picture because it shows you kind of what's moving when and where. And so if you look at the blue is reabsorption and the green is secretion. Okay, so here's our blood supply, here's the glomerular capillary, here's the glomerular capsule, here's the proximal convoluted tubule, here's the loop of Henle, distal convoluted tubule, and this is the collecting tubule. And so if you go through and you can see what's happening in each part of the nephron. Now again, I'm not going to read all this to you because you've got this that you can look at in your book, but you need to know this. You need to be able to tell me what is moving when. So in the proximal convoluted tubule, you have both reabsorption and secretion are occurring. Okay, you got reabsorption of the filtrate volume, mostly sodium and water, bicarbonate, glucose, amino acids, other nutrients, but you're also getting rid of hydrogen, ammonia, and some drugs. So like we do a drug test or urine test, this is where some of that's going to be secreted into your urine. As you go through the loop of Henle, mostly you're looking at water, salt, and water balance. You see absorption of water. On the descending side, the descending limb of the loop of Henle, you see ion reabsorption on the ascending side. That's important, so make sure you pay attention to where it's occurring, ascending or descending limb. You also see the secretion of urea into the ascending limb of the loop of Henle. In the distal convoluted tubule, both absorption and reabsorption, absorption and secretion. You have things like sodium and chloride and calcium, which are under hormonal control. You have potassium secretion, which is again under hormonal control, aldosterone. If you look at the collecting tubule, again, both absorption and secretion. You get water absorption, sodium absorption, urea absorption, again, all of these by hormones, under hormonal control. And then secretion of potassium, again, by hormonal control. All right. The last thing that can happen in the collecting tubule is, depending on what's going on, kind of depending on where you are in your homeostatic balance, you can get either reabsorption or secretion of things like hydrogen, bicarbonate, and ammonia. And these play a role, again, in that acid-base balance. So this is a great picture. I get a lot of questions on my exam from this particular picture. All right, so that's it. So we've introduced urine formation, how it occurs, reabsorption, filtration, reabsorption, and secretion, where that occurs and what part of the tubule, and what's actually being secreted and reabsorbed. Okay, so that's it for now, and we'll pick it up on the next video. Thank you.